All right, so we're going to begin the skeletal system. All right, so skeletal system functions provide support, framework for soft tissues and the muscle attachment. Generally, what we think about is the architecture of your body, the hard architecture, that can withstand gravitational forces and for muscles to attach to. And then when muscles shorten and lengthen, it moves the skeletal system. Protection, so it has bones that surround the brain, the spinal cord, the heart and lungs, like your ribs and your sternum. Movement together with muscles, move the body. Mineral homeostasis, so we store lots of calcium and phosphorus. Hematopoiesis is blood cell production from red bone marrow. And storage of fat, triglycerides, as yellow bone marrow. Okay, so we have, when we start looking at the skeletal system, we start looking at the cartilages, and we were talking about that, in the last, or the second to last chapter we covered before the exam one. We were talking about tissues, and we said of the connective tissues that had the most variety, and the cartilages we would be looking at when we look at the skeletal system. So now we're going to do that. We're going to look at hyaline cartilage, elastic cartilage, and fibrocartilage. And the blue, green, and red here are the colors on the subsequent picture, the areas of the body identified as blue, green, and red. We have two types of bones. We have compact bone and spongy bone. All right, so here we're looking at the skeleton. In the center where we have the darker shaded bones, that is considered, or that is the axial skeleton. Those are the bones sort of in the center of your body, your ribs and your sternum. And your appendicular skeleton are the bones in like the lighter vanilla color kind of appearance. Right? Those are the bones of your extremities and the bones that attach those extremities to your axial skeleton. So that would include each side of your pelvis connecting your legs and your sternum, and your clavicle and your scapula, attaching the bones of your arms to the axial skeleton. Then we have some examples of locations of hyaline, elastic, and fibrocartilage. Hyaline cartilage is the light blue, and I will tell you also that most models that you look at in lab, if it's hyaline cartilage, it's going to be a light blue also. It just seems to be the color code that most model makers and design artists, creating pictures, use for hyaline cartilage is light blue. Elastic cartilage locations, there are just a few of them: ear, epiglottis, and eustachian tube, we asked you to know. So up here on the ear. Fibrocartilage locations are areas where you have a lot of impact and stress. So between the vertebra of the back called intervertebral discs, the pubic symphysis, here where the two sides of each pelvis come together anteriorly, and then the menisci of the knees, we have kind of brake pads or cushioning between the long femur and the tibia bone of the long extremities, the lower extremities at the knee joint. So hyaline cartilage is the first cartilage. It's the only collagen that contains collagen fibers that are not directly visible microscopically. So there are some fibers there, but we generally don't see them. I've never seen a picture of hyaline cartilage that actually had the collagen fibers, evident. It is the most abundant cartilage, and its locations are the most numerous as well. So these are located at the ribs. They're not the ribs themselves, but it's the cartilage that connects the ribs to the sternum, called costal cartilage. Regions of the trachea and the majority of the trachea, right? The larynx, nose, articular ends of bones, of long bones. When we look at these bones, we're going to see that, especially where we have synovial joints, which are the majority of our joints, the ends of the bones that meet together that have to move past one another repeatedly over and over and over, are going to have cartilage there, and it kind of keeps the bones from fraying. And then we have what's called the epiphyseal plates. Those are areas bone elongation when we lengthen bones. We'll look at those. The cells that produce cartilage are called chondrocytes, right? Chondro for cartilage and site for cell, chondrocytes. So here's a picture of hyaline cartilage. Sometimes it will be kind of a light lavender background. Sometimes it'll be kind of a light pinky or kind of a background. Depends on the stain that's used. But generally, you see chondrocytes sitting in their lacunae. Those are the cells. Their nuclei takes up a dark pink, dark kind of black kind of a color. The space they sit in is called the lacunae. And the material in between each lacunae that kind of houses an osteocyte is the matrix. So it's generally devoid of visible collagen fibers at all. The material between each cell and its lacunae is generally kind of pale, staining, translucent. So ding, 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 that's going to be your big visual key, that you're looking at, hyaline cartilage. This picture is generally coming from the trachea. It shows you an area, costal cartilage, but these are, frequently the ones we look at in lab are going to be from the trachea. So we use these same slides in the respiratory system in A&P2 when you look at that, but it's an excellent example of hyaline cartilage for the A&P1 labs. From a distance, it kind of has a soap bubbly appearance, with absence of fibers. When you see the subsequent cartilages that are very abundant in fibers, you'll definitely see the difference. And if you are taking even a lab online versus a lab face-to-face, it's good to group these slides together, and if you can, in lab, to have 3 different microscopes set up, one looking at hyaline cartilage, one look at fibrocartilage, and one for elastic cartilage. And you kind of move back and forth between them all so that you can really develop an eye for, which is which. So if there are any study rooms open, that would be good to do, or with a tutor if you're meeting with any of the tutors. You develop an eye for it very, very quick to at least identify the difference between them. Right, so our next cartilage is elastic cartilage. It contains primarily elastin fibers or elastic fibers and collagen a bit. It has what we call the Scooby-Doo eyes appearance because the chondrocytes, when they sit in their lacunae, it has a very pale environment to the chondrocytes in their lacunae. It's very translucent. But the dark fibers, elastic fibers, they take up a lot of stain. And so they're very dark fibers in contrast to the chondrocytes sitting in their lacunae. And, this is just from a reference that another instructor would use, or another anatomy professor, because he watches a lot of cartoons, said it looks like Scooby-Doo peering out of the dark. Locations are ear, epiglottis, and eustachian tube, or more specifically, external ear. External ear, epiglottis, that is the flap of cartilage tissue in your throat, in your larynx, which holds your voice box or your vocal cords. And that flap of elastic cartilage closes off that airway entrance when you're swallowing food, so food doesn't go into your airway. And the eustachian tube, we'll see again when we look at the special senses models of the ear. It connects the middle ear, which is the region on the other side of your tympanic membrane, to the back of or let's say that upper part of the back of your throat, which is called the nasopharynx, and it helps balance air pressure. But you'll get to know some of these things as we progress through A&P1 and A&P2. And the cells, of course, are chondrocytes. So here we have, this is the appearance of elastic cartilage. We can see, and it's magnified more than the hyaline cartilage. So here we have the chondrocytes sitting in their lacunae, their little room, right? And then you see these fibers. Now, generally, you will even see more fibers than that, so that it's very dark purple regions all between these chondrocytes in the lacunae. So compared to the hyaline cartilage, that material between each chondrocyte, is very pale staining. This will be fibery, dark staining fibers. So that's a visual cue that you're looking at elastic cartilage. Fibrocartilage contains chondrocytes as their cells, but their fibers are mostly collagen fibers. And they produce what I call the Van Gogh appearance, where these collagen fibers are very parallel and they're wavy. And they take up this light blue stain. The chondrocytes, their nuclei will take up like a bright fuchsia color. So it's really a rather pretty specimen. So you have these light blue, wavy, very dense, regular collagen fibers, and periodically, depending on where you're navigating through the slide, you'll see a few or more bright fuchsia-colored nuclei, which are the nuclei of the chondrocytes. Now, collagen fibers withstand tensile strength, which means they withstand compression, being squashed essentially, instead of pulled apart and rebounding again, like an elastic, or elastin fiber would do, like an elastic band, stretch it and it bounces back, right? So these are very high compression areas, intervertebral discs, pubic symphysis, and the menisci of the knees, or the meniscus. So this is an example of what fibrocartilage looks like. So you see all those light blue wavy lines? They kind of remind me of Starry, Starry Night by Van Gogh. Have you ever seen that painting? And you can see that bright fuchsia dot there. That is a chondrocyte sitting in lacunae. Right? They're rather abundant in this particular example, this picture for the textbook. When you're looking in lab, you kind of have to navigate around a little bit. But clearly this appearance, if you can set up microscopes, if you're really doing this face-to-face, and if you're not, and you're studying from home from an online class, make it, a PowerPoint of like all the histology, and put them like I do, like the PowerPoints in slideshow mode. And you can have one with the next, one versus the other, but the other, or one picture, and you can, one slide with all three pictures of your different cartilages. And then the next slide would be, which one is, A, B, and C, or which is in the upper left and then the upper right and the lower. And you keep looking at them to develop, your eye for it, because they are quite different from one another. And so is compact bone and spongy bone. So when it comes to histology, this is actually rather, I think, easy to distinguish between, at least identifying what tissue it is. And then from there, you have to know, well, what would I expect to find in this tissue? Like this is fibrocartilage. Well, what would I expect to find there? Collagen fibers and chondrocytes in the lacunae there. Okay, then we move on to compact bone now. So those are our three cartilages. Compact bone is arranged in osteons. The osteons are the repeating structural unit of compact bone. Kind of looks like a tree that you cut down and you look down on it, and it's all the rings of the tree with a central, region to it. And they repeat all over through the compact bone. Compact bone is the outer rim of all these long bones and the outer rims of even irregular and flat bones. And I'll show you what they look like in a couple of pictures. Mostly what we study are the long bones, but bone is not solid throughout. There's a large, depending, like a long bone has a large cavity in the center called the medullary cavity. And even a flat bone, it has compact bone to the outer margins of it. And then in between the two, kind of like a sandwich, two pieces of bread would be the compact bone. And in between the two pieces of bread would be where we find spongy bone. So I'm going to show you pictures, but I'm just saying this, when it comes to bone, especially bones of your body, they are not solid throughout. The compact region of it is to the outer margins. And then deeper to that is going to be maybe fat-filled cavities or cavities filled with red bone marrow, such as spongy bone, and some hard substance in there, but it's not solid. So even the compact bone regions are not solid. As we look at this, you're going to find that these interwoven cavities between them. We do have in between these rings of compact bone, or rings of what's called osteon in compact bone. We have collagen fibers spiraling through there, and that gives the bone a certain amount of flexibility. Oh, sorry, I should have put my phone on mute. Okay. The cells we find in bone. We have osteocytes, osteoblasts, and osteoclasts. It's a little more complicated than cartilage. Osteocytes maintain bone matrix. Osteocytes will be found in compact bone territory, right? in the lacunae there, and then in some lacunae in trabeculae. Trabeculae is one of the two components of spongy bone. All right, then we have osteoblasts and osteoclasts, and they work rather in opposition to one another. Osteoblasts build up bone found in the periosteum and endosteum, and those are membranous layers, membrane layers to the inside and outside margins of our compact bone. Osteoclasts break down bone, found in the same place that osteoblasts are, found at periosteum and endosteum. So they work in opposition to each other. They're going to be stimulated by different hormones. And they are in higher populations. Let me just say, they're not necessarily equal amounts of osteoblasts and osteoclasts at the periosteum and the endosteum. We generally have more osteoblasts to the outer margin, the peri, and more osteoclasts to the endosteum region, but some of each are generally found there, just in different percentages. Okay, so here is a picture of compact bone. I will say that the slides in lab that we use are very similar to this. They're not exactly like this, but they have the same appearance. Do you see how you have, it looks like trees and rings of a tree with a dark center and then another smaller tree next to it. You can see multiple rings to it. And then down below, you have another ring of trees. That ring, with all its individual, or that tree, I should say, with its rings, with the dark center, that is one osteon. And they repeat all over throughout compact bone. And so that one tree and all its rings is one osteon. In the center, we have what's called a central canal. Each ring is called a lacunae. Where you see those dark slits, sorry, each ring is called a lamella. The dark slits where we find the osteocytes are the lacunae. Sorry about that, just like it would be in cartilage. And you can see these little fine passageways that take up some darker brown stain that are radiating from the central canal outward. These little fine, little dark brown lines there, they're called canaliculi. So we'll talk about that microscopic anatomy and what functions those impart in just a moment. All right, the chemical composition of bone. We have organic components and inorganic components. Sort of like when we were looking at the connective tissues, we were talking about the cells and then everything, all the matrix found between the cells. Well, we have, of the organic portions, we have cells and then osteoid. The cells of bone are either osteoblasts, osteoclasts, or osteocytes, we just described. The osteoid is the organic matrix, secreted by osteoblasts that contains what's called ground substance, which have lots of proteoglycans and glycoproteins, so combinations of carbohydrates and proteins, and a lot of collagen fibers that give bone a certain amount of the compact bone flexibility. You want it to be firm, but you don't want it to be kind of dry and fragile, so it just snaps. So, there is a certain amount of flexibility of bone. It doesn't feel that way. It doesn't feel flexible like, if you move your ears around back and forth, and the tips of your nose, clearly that is flexible. It doesn't provide that kind of flexibility to that degree, but it does from each, between each ring of lamella in each osteon, have collagen fibers there to withstand a lot of compression, and it gives bone a certain amount of bendability before it snaps. The inorganic portions are the hydroxyapatite. Those are our mineral salts, around 65% of the bone mass. We're looking at mainly calcium phosphate crystals, and it's the part of the mineral salts, it's what gives the bone, the matrix of it, the real hardness, hardness and resistance to compression. Okay, now we have spongy bones. We have two major types of bone. Compact bone is what we mostly think of with bone, but we have lots of spongy bone, and that's found deep to areas of compact bone. So it's composed of two major portions, the trabeculae, which are the walls of dense tissue or walls of dense tissue that give it its tensile strength, so it resists compression. It's called, it looks like walls of a sponge. That's why we call it spongy bone. But in life, you will find material between the walls of the sponge called red bone marrow. And this is where all blood cells originate, all blood cells. So when we see a picture of it, it'll make a little more sense. Okay, here we're looking at a flat bone. So up in the upper left-hand corner, you have a skull, and it has a little square from there because that's a great example of flat bones. Right? And they're magnifying it below. All right, so the bones, these flat bones of your skull, have an outer compact bone region. So you see that to the top and then to the bottom. And in between those two layers is spongy bone, or what we refer to as diplo. Right? So you see how it looks kind of like a honeycomb appearance? Yeah. Well, when you just have this bone and some, it's not live tissue, and the red bone marrow has evaporated and been replaced with just air. Right? So think of even just a kitchen sponge, where you have walls of the sponge, which is usually rubber, and then you'd have air in between. Well, that's sort of what it looks like. You have compact bone to the top and to the bottom. So it's a compact bone sandwich with spongy bone in between. So the part you can see there, which are the walls of the sponge, is the trabeculae, and it's magnified below. Right? So these extensions and kind of finger-like projections of hard trabeculae course in different directions, like the walls of a sponge. Right? And so down on the bottom, you can see they travel in different directions. And that's what provides, even though it's not really solid like compact bone, it provides a lot of strength to the bone. In life, you would find red bone marrow all between those. So the slide over to the right-hand side gives you a little better appreciation of that. The pinkish staining material in the center in the shape of a Y, that's your trabeculae. And you can see some osteocytes sitting in the lacuna. But the material between those two, the lower right and left into the upper region, you can see that the dark purple staining granular material, that is the red bone marrow. And often, as a part of red bone marrow, depending on the location, you will also have fat cells. So those other look like water balloons kind of there, those are adipocytes. And the ratio of red bone marrow to adipocytes can change depending on the location, of the spongy bone. But here you can see some red bone marrow, even though it's stained purple, it's red bone marrow that produces all blood cells, red blood cells, white blood cells, and platelets, and some fat cells in between. Okay, so we're going to talk about bone shapes. This sometimes is part of one of the exercises for the skeletal system. We look at long bones. Long bones are longer than they are wide, with a shaft and two ends. This includes all the limb bones, the long bones of your arms and legs. We have short bones that are cube-like bones, mostly composed of spongy bone, sort of the appearance that we just saw before, but they're not going to be maybe completely flat like that. They'll be a different shape, but again, compact bone often to the outside and spongy bone to the inside. This includes a lot of the bones of the ankles and the wrists. They're very unusually shaped. You'll see that as we cover the appendicular skeleton. And sesamoid bones. Sesamoid bones are bones that grow in tendons and muscles, and they vary in size. We have a lot of them around the ankles, to the inside and the outside of your ankles and the feet, and some around your wrist. And the largest one is your kneecap or patella. That's a sesamoid bone, the type of short bone. Right, then we have flat bones, like the one we saw of the skull. So a lot of the skull bones, your sternum, your breastbone, and ribs, they're very thin and flat, often with a slight curve, and it resembles a sandwich of compact bone and spongy bone in the center. Then we have irregular bones. And a good example of irregular bones are the vertebra of your spine, all different shapes and sizes. The three bones that come together and fuse to form each side of your hip, each side of your pelvis, I should say. The hip is sometimes referred to as the hip, is composed of three bones, like the ilium, the pubis bone, and the ischium bone. And they are very irregular bones. Right? So here we're back to our skeleton. They give you an example of, we'll start with a long bone. That's a humerus. Right? So the long bone of the humerus of the lower part of the forearm is the radius and ulna. The long bones of the lower extremities, the femurs, tibia, and fibula. Irregular bones are the bones of the vertebral column here, and to each side of your pelvis. Flat bones such as your sternum, and short bones such as the bones of your ankle, which are called tarsal bones, and of your wrist. You have two rows of four that are all unusually shaped, and those are short bones as well. Okay, so we look mostly from here on at long bones. We're going to look at the gross anatomy, which is the anatomy, the large anatomy of a bone. And we have these different regions. We have the shaft of the bone called the diaphysis, and the knobby ends of the bone called the epiphyses. So the diaphysis is the long axis made of a thick collar of compact bone surrounding what's called a medullary cavity that contains either red bone marrow or yellow bone marrow, depending on the age of the individual. And then we have the ends, composed of compact bone and spongy bone to the interior. The joint surfaces of these epiphyses, or the ends of the bone, are where we find articular cartilage, which as a structure, it's called articular cartilage. The tissue that forms your articular ends of bone, articular cartilage, is hyaline cartilage. So as a structure, it's called articular cartilage. The tissue is specifically hyaline cartilage. And then we have on lung bones, depending again on the age of the individual, either an epiphyseal line or an epiphyseal plate. The epiphyseal plate is a region between the diaphysis and the epiphysis that has hyaline cartilage, so that the bone can lengthen and become longer and longer and longer when you want to grow your skeleton, when you're trying to increase the length of your entire skeleton. It occurs, until you have late adolescence or early adulthood, when these fuse down, and now you've reached what we call skeletal maturity. You've grown to the height that you're going to get to, and then the bones will no longer lengthen. And that occurs at what's called the epiphyseal plate. You'll have an area of hyaline cartilage there. And during especially growth spurts, during adolescence, they will grow maximally, longer the bones and wider, and then they'll be replaced. That area of hyaline cartilage will be replaced by compact bone, and it goes from being called an epiphyseal plate to an epiphyseal line. All right, so here we're looking at a humerus, a long bone. Look where they identified the epiphyses. So the knobby ends of a long bone are called the epiphyses. The one that's closer to the point of attachment of the entire arm or leg, to the shoulder joint and hip joint, is the proximal epiphysis. Then you have the shaft called the diaphysis and the distal epiphysis. Okay. So now that we're looking at this entire long bone, you can see the outer kind of solid vanilla color region is the region of compact bone. And then deep to it, you can see sort of that soap bubbly honeycomb appearance. That's your spongy bone. The epiphyseal line is a solid region between those two epiphyses that used to be your area of elongation, and now you've reached skeletal maturity, and it's replaced with a solid bone called the epiphyseal line. And that happens at both ends. In the center of the long bone, we have a medullary cavity. And we'll see another picture, like over to the right-hand side, you can see that medullary cavity there. In children, that will be filled with red bone marrow. As an adult, it will be filled with yellow bone marrow. We have blood vessels that enter at different points and branch off, right? That becomes the blood supply within each of those osteons of the compact bone. Each of those osteons, the tree with rings of the tree, has its own blood supply. So we're going to look at, this anatomy in more detail. It's also pointing to here, the outer surface of that entire long bone has a membrane there called that periosteum. And to the inside margin of that rather thin, comparatively speaking, rim of compact bone is going to be your endosteum. Okay, so if we go, that's, look at a lot of the gross anatomy at, image A. B is kind of up to the upper rightish corner. We take a region of that bone and we magnify it. And now you get a little better idea of where the periosteum is. It's kind of a light bluish layer to the outside. You can see a region of compact bone. We're looking down upon it and then, kind of in a sagittal view, so to speak, seeing what it looks like if we could see it, watching the vertical portions up and down from superior to inferior. You can see a big blood vessel in there. And then as we radiate from the periosteum to the spongy bone-ish portion, we have an endosteum there, right? It's just cut, the color of it is a darker brown. See the lines pointing to the endosteum. On our models, the periosteum, the endosteum will be the same color, like a grayish color, so that okay, one's to the outer margin, one's to the inner margin, periosteum versus endosteum. But you can see a little bit of spongy bone in here as well. And then if we're C is, we take a cross section and we look at, okay, what does it look like when we kind of cut into it and we look down on it? So you can see that that compact bone is circumferentially around, the long bone, but in the center, we have this cavity, and that's your medullary cavity. You can have the outer connective tissue with the periosteum. The endosteum would be to the inner margin between the fat that you're looking at there, the yellow bone marrow, and the compact bone, this kind of that dark brown kind of layer there, that would be the endosteum. Okay, so bone membranes. We have a periosteum as a double layered membrane covering the diaphysis, the outer portion of the bone. This is a very good surface, for being the outer connective tissue layer of an organ. You're going to have this repeating throughout every system, but the outer connective tissue layer, or let's say every organ has an outer connective tissue layer, and they all serve the same purpose. Not exactly the same surface, because some things are unique in the location of this, but I'll give you an example. That outer connective tissue connects to the compact bone itself, all right? It helps anchor blood vessels and nerves that have to enter and exit this compact bone, right? Because all this is living tissue. Even though it looks hard and dense and it looks like it's completely solid, it's not. There are living cells in there. And you have to have a nerve supply and a blood supply and a lymphatic supply, right? So that outer connective tissue helps secure the blood vessels and different nerves and lymphatic structures as they enter and exit the bones, so they don't get pulled or torn. But on compact bone, it is especially a good surface for the tendons of muscles to attach to, so that when the muscles contract and they move the bones, that the tendons are actually attaching to this connective tissue of the compact bone, not the compact bone itself. The periosteum and endosteum also serve another function. It's where we find osteoblasts and osteoclasts for what's called bone remodeling and growth in diameter. All right, so bone remodeling means you are going to use osteoclast when necessary to maybe break down some bone, and osteoblast to build it back up and maybe repair some bone. The endosteum is the internal membrane lining the medullary cavity. It lines the medial layer of the compact bone or the medullary cavity, and also contains osteoblasts and osteoclasts for bone remodeling. They're just in kind of different percentages of populations, generally for the the endosteum versus the periosteum. All right, then we have short, irregular, and flat bone, gross anatomy. That is that, again, the compact bone sandwich I was talking about, compact bone to the outside and inside, and spongy bone in between, where that spongy bone is often referred to as diplo, just a fancy word for spongy bone there. Okay, then we're going to move on to hematopoiesis. So what does that mean? It's the formation of blood components, right? Formation of blood components, especially red blood cells. So all blood components come from red bone marrow. It's not just red blood cells. It's red blood cells, white blood cells, and platelets. In children, red bone marrow is found in the medullary cavity of long bones, in the humerus, in the sternum, and really in every available spongy bone cavity area that there is. This will make sense in a moment, right? So wherever you can in children, wherever you have these cavities, especially in long bones as well as flat bones, you're going to have a lot of red bone marrow there, making lots of blood cells. In adults, once you've reached skeletal maturity, the red bone marrow in the medullary cavities of the long bones become replaced with yellow bone marrow. And yellow indicates fat. Yellow bone marrow is fat. And so we're, it's not necessarily, we're replacing that red bone marrow with yellow bone marrow to store energy, but it's a good tissue to put in there that is rather benign. So it kind of, think of any function associated with fats. From the chemistry lab or the chemistry chapter, cushions and insulates organs, long-term energy storage, things like that. But the bottom line is that children, because they're increasing in size, the volume of blood that must be circulating is going to have to increase, on a regular basis, not necessarily profoundly, except maybe in adolescence, but you're going to have to increase the amount of blood circulating. So let me give you an example. The average volume of an adult, the volume of blood circulating at any point in time is about 5 liters, give or take the size of the individual. If you have a really tall person or large person, they're going to have more blood. But adult blood, you just want to think about 5 liters. Well, you don't have the same amount of blood circulating in an infant because they're just little dudes or dudettes. There's, you maybe at best have a liter, okay? So, but as the child grows, the amount of blood required to be circulating needs to increase. And red blood cells only survive about 120 days. So your body, the red bone marrow, the majority of the red bone marrow there, as an adult, or when they're very, very small like this, because they're not growing rapidly, is really there to replace the red blood cells that are dying off every 120 days. But very quickly, children start to really grow. And as they're growing, they need to not only be replacing what's dying off every 120 days, which is about 1.5% of any of the red blood cells that are in circulation at that point in time. They have to be always increasing the overall volume, from 1 liter to 1.1 liters to 1.2 liters to 1. So they need to have more areas in bone designated for bone cell production, factory areas for red blood cells, and all the other white blood cells they're going to need as well, and the platelets. And so as they get larger and they grow, you have to have lots of factories putting out this, these blood components. But by the time you get to skeletal maturity, which is generally late adolescence, early 20s for some men, men, they have your growth spurts sometimes longer before those epiphyseal plates get replaced with epiphyseal lines. You need to have a lot of blood being produced when you're growing, especially rapid growth. But once you get to that point, you just really need to maintain the status quo, so to speak, the 5 liters, give or take, with exception. I mean, sometimes you have bleeding and you have, all right, that's different. You're really going to pump up how many red blood cells. And that's going to be the job of a hormone called erythropoietin that we look at in AMP2. But generally, you just have to maintain what's there. So you can work off of fewer areas of red bone marrow, and a lot of those will be in the fat. I keep saying fat bones, flat bones of the body, and it's some of the epiphyses of the long bones. I hope that kind of makes sense. The difference between some of the requirements, the metabolic requirements and changes between infants and children's and adults when you're not going to be growing much more. Okay, so now we're going to look at the microscopic anatomy of compact bone, the osteon, the repeating structural unit of compact bone. All right, the rings, there's a lot of terminology in every system. All right, so the rings that form the osteon are called lamella. They're just called lamella instead of rings. The big channel in the center is the central canal, or sometimes referred to as a Haversian canal, that contain blood vessels and nerves. So they travel in a vertical direction. We have the lacunae, which are the little spaces that house an osteocyte. They're generally between lamella to lamella. We have osteocytes that maintain the bone matrix, right, that are found in this compact bone territory. They're in their lacunae. And then we have canaliculi, or can I lick your eye? That's how you gotta remember it. These are very narrow passageways. Large passageways are often canals of some kind. That will allow blood vessels and nerves to pass through. But when they're a narrower pathway, this is really for diffusion of material, and that's what a canaliculi is. They connect and radiate kind of outward, back and forth from the central canal and connect to lacunae to lacunae to lacunae, from the center going all the way out to the periphery of that osteon. And that allows nutrients like oxygen and glucose and maybe some hormones for chemical communication to go from the arterial blood to diffuse down its concentration gradient, where it's high concentration in the arterial blood that feeds and travels out to every lacunae and the osteocytes that sit there and live there to give them nutrients. Those cells are alive. In return, the waste and the metabolic waste products of cellular activity of the osteocytes will travel in the opposite direction from where it's being generated in high concentration in the cells, through the canaliculi back to the venous blood supply, the blue vessel, and be swept away. So those canaliculi connect the osteocytes with the central canal for transport of nutrients and wastes. So here we're looking at, we cut into a long bone. This is a femur, all right? You can see the periosteum to the outside. You can see the compact bone region. Right? The osteons are looks like a tree and all the rings of the tree. Right? They're called, we have different types of rings of compact bone tissue. But the rings we have, circumferential lamella are the rings of compact bone that go around the entire circumference of the bone. And then we have what's called concentric lamella, which are the lamella within one osteon, just in case you see those terms and you're wondering what that means. Okay, so lamella just means rings of, of matrix, the hard part that you're looking at here. You can see as we look down the long shaft of the diaphysis, you see how those, what would be the central canal of a couple osteons that are cut on the right-hand side here. You can see how those vessels run vertical. You have connecting points where they can branch off. So there's sort of an internal road system for arteries to enter compact bone and ultimately branch off in different ways to provide a blood supply in the center of each of these osteons. So if you look in each of them in the center of the ones, here in this compact bone region, you can see a red vessel, a blue vessel, and something yellow, and that's a nerve. So each central canal provides the nutrition for all the cells and osteocytes that are part of its osteon. Right? These are, of course, histological slides at the same. We're looking at the one over to the right-hand side is a little easier to visualize. That's one osteon. There's an adjacent one to it, to the left-hand side. But you can see kind of the rings of the lamella, the central canal. Looks kind of bloody in there, and that's because it would be, because you have dried blood from the blood vessels being cut, when you made this, and this slide, this tissue slide. You can see the rings of lamella. You can see these dark kind of ovals. It looks like black little areas, slits that are between lamella, and that's an lacunae where you would find an osteocyte. And then you can see those little, it looks like fine lines radiating outward from the central canal outward toward the rim or the outer margin of that osteon. And those are the very narrow canaliculi passageways, for diffusion of nutrients, including gases, from the blood supply in the central canal out to the osteocytes in their lacunae, and from the lacunae and the osteocytes back toward the blood supply. It just depends on what the substance is. So we can have bidirectional diffusion of different substances. That makes sense. Okay, then we have a Kahoot. So let me stop my share and then reshare as soon as that kicks in. It doesn't seem to be working. Hold on now.
Am I the only one that doesn't see anything?
No, me too.
Me too.
3. Does this mean we can like leave?
Did anybody get 100 on the test?
I got a 98. I think I missed one.
I don't know which one I missed, and it's driving me crazy.
I know. I wish you'd release it so I could tell.
I got a 98, and I'm super pissed. I know I got them all right.
You think so? All right.
You
Well,
pieced that in 98. What?
I'm heading out. Would someone send me a message if she gets back in here or something? Unless everyone's leaving, in which case.
Class is over.
All right. I'll see y'all next time.
Yeah, I'm gonna dip. Bye.
Later.
I don't know what happened. Sorry about that. It like trying to open up that Kahoot quiz like crashed my computer. And I just downloaded about 3 weeks ago that Microsoft 10, which I hate because then it takes you 10 minutes to reboot your computer. It's god awful. Anyway, so what I'm going to do is hopefully, oops, and see now it's just switching different pages. All right, is I am going to try to, if you guys can hear me, I'm going to switch back and forth here because it's opening different pages for me. Let me try to get this to, there we go. All right. I'm going to continue. I'm going to open up the notes again and try to move on, but I'm not going to open up that Kahoot quiz. I unfortunately, and I won't do this again, is if you could hear the dogs barking in the background, I'm home doing this. And this is one reason why I don't usually do that. And I got a little lazy today and I was like, oh, let me just Zoom from home and I will never do this again because you just never know when I've got a laptop here, whether I'm going to have a problem. Anyway, so what I'm going to do is I'm going to open up this PowerPoint again from my thumb drive here. And I'm going to reshare, and we're just going to continue with the skeletal system. So if you're just patient just a moment, let me just open this up and then I'll reshare it. And we'll get through probably, maybe not the whole thing, but the majority of the skeletal system. And then we will look at whatever is remaining in axial and appendicular skeletons on Wednesday. Okay, so there's that. Right now I just have to reshare it, hopefully. Yay, I think we're making progress. So sorry about that. Boy. Okay. So does everybody see the notes now?
Yes.
Okay, just so I get thanks for the feedback. Good. Okay. Oh, brother. All right. Well, that was fun. All right, let's put this in slideshow mode. And let me just start here and work my way through really quick. So we did a lot of that. Yay. Okay, the god awful Kahoot. All right, we're going to move on. All right, so now we're going to talk about formation of bone, which is called osteogenesis. And we have two major methods that you form bone. You either do it through what's called intramembranous ossification or endochondral ossification. And that's our bone lengthening. Bone growth, all right, is in two directions, so to speak. We generally, especially long bones, we're primarily looking at long bones. We have to grow bones in the length, and we also have to grow them in diameter, which is called appositional growth. So when you're small, you have, obviously, infants have very small bones. We don't want to just lengthen them, otherwise we'd have these toothpicks for bones. We need to increase their diameter as well. And so in doing so, it's called appositional growth. We thicken up the compact bone rim a little bit, but we increase the medullary cavity in the center even more so that it's not super duper, thick compact bone regions. But I'll show you, what we're referring to in that. Okay, so we have what's called intramembranous ossification. This is how flat bones form, like the skull and the clavicles. It develops from and originates from a different type of tissue. This develops from fibrous connective tissue. Usually begins in the center of a membrane called the ossification center. But its origin is fibrous connective tissue, and it develops into those flat bones. Long bones do it a little bit differently. We develop the bone. We have a primary and secondary ossification center, and then called endochondral ossification, and then we have elongation of bones. Long bones develop from the hyaline cartilage model, it's called. Instead of being fibrous tissue, this is hyaline cartilage. So kind of picture kind of a large droplet, so to speak, of hyaline cartilage. And that's where how these long bones will develop from embryologically. They start by kind of looking like a big milk bone, so to speak. And the chondrocytes in the lacunae in the hyaline cartilage are getting their nutrition through diffusion from the tissues and the fluids and things surrounding it. So all those chondrocytes in the lacunae of the hyaline cartilage are getting their nutrition through diffusion from surrounding area. But eventually, the center region starts to die off and becomes replaced with compact bone, and encourages blood vessels to grow into it and supply a blood supply to now bone cells of compact bone. That first region that goes through these change is called the primary ossification center. So I'm going to show you a picture in just a minute. After that develops, and the whole structure in the meantime is getting larger and larger and larger, the ends, which ultimately become the epiphyses, go through this same kind of metamorphosis of being hyaline cartilage and being replaced with compact bone and spongy bone. And those are the secondary ossification centers. The primary ossification center at the end becomes the diaphysis of the bone with a medullary cavity. And the secondary ossification centers, end up being the epiphyses of the bone with compact bone, in a spongy bone in the center. And then the remnants, hyaline cartilage, are at the articular ends of bone in light blue, and are found at the epiphyseal plates for bone elongation. So you start out entirely with hyaline cartilage, and then you replace that with as much bone as possible. At the end of ossification, hyaline cartilage remains as the articular ends of bone, often indicated in light blue, and the epiphyseal plates, the junction of the diaphysis and the epiphysis, where bone elongation will occur. All right, so this takes us to the image. There really should be another image to the far left-hand side preceding what's called the bony collar formation, where you see just all hyaline cartilage. So although this picture isn't, it gives you a pretty good indication, the hyaline cartilage should have this soap bubbly appearance to it, like all hyaline cartilages do, and it's in light blue. All right. So as this gets larger and larger, the hyaline cartilage, region becomes larger and larger. You start to have a change to essentially the cells in the center of the hyaline cartilage start to die off because the diffusional pathway is too extraordinary. And the chondrocytes essentially start to die off and they start being replaced by bone cells. And that is your bony collar that forms here. Kind of the waist area of this big hyaline cartilage milk bone. So that is the primary ossification center. So in image 2 here, cartilage in the center of the diaphysis calcifies and then develops cavities, and that will ultimately become the medullary cavity. It encourages in 3 blood vessels to bore through there and branch off. Thus, now we are developing the blood supply to the center of each of the osteons that's developing there. And look, it's getting larger and larger and larger all the time. So as it becomes even larger and we're done with modifying the primary ossification center, now the ends of this hyaline cartilage model goes through the same process the primary area did, where you the cartilage cells start to die off and they're replaced by bone tissue. It encourages blood vessels to grow in there. Those are the secondary ossification centers, the second area to go through change. And it grows larger and larger until finally it's about the long bone size that you see here. The remaining light blue areas are the remnants of all that hyaline cartilage at the articular ends of the bone and at the epiphyseal plates. And now it starts to look like a nice long bone. The epiphyses, proximal and distal epiphyses, and the diaphysis. And that's how long bones develop. Okay, we need to increase the length, which occurs at those epiphyseal plates. So let me go back a picture real quick. So you see those epiphyseal plates where you have hyaline cartilage. You have the proximal end of it and the distal end of it. Do you not? One edge of it is articulating with the epiphysis and one side is to the diaphysis. And the same with the one, at the distal, so to speak, epiphyseal plate. One edge of it is closer to the diaphysis. One margin of the light blue is closer to the epiphysis. When the bones elongate, they march kind of and extend away from the diaphysis. So on the epiphyseal plate, to this superior aspect here, it increases and it pushes forward the hyaline cartilage region upward and toward the articular cartilage up at the top, thus extending the length of the diaphysis. But the same thing happens at the bottom too. You just extend in the different direction. So the hyaline cartilage will push kind of forward toward the articular cartilage at the inferior aspect of that long bone. And by doing so, keeps increasing the length of the diaphysis or the shaft of that bone and remodeling along the way. So we increase our cartilage region and it gets replaced with compact bone, and then your osteoblasts and osteoclasts will kind of shave it down a little bit. So it keeps that diaphyseal area, the shaft of the bone, looking like normal diaphyseal bone of long bone. It's kind of hard to describe, but I'll show you in just a minute what the remodeling is going to look like. Okay, so we find these 4 zones of tissue going through this process of being converted, so to speak, from cartilage to some kind of compact bone. And they're in the same, I'd say, sequence of zones from resting cartilage to calcifying matrix. Right? So you have the first region is called the resting cartilage or resting cartilage of hyaline cartilage. You're going to have one region of it that looks like hyaline cartilage, that light blue cyclophoid kind of material. Then the next layer is called the proliferating cartilage, or the zone of proliferation, where the cartilage cells are going under high mitotic division and stacking on themselves. So it's going to have what's called a stack coin appearance. So your zone of proliferation means the numbers are increasing, proliferating, increasing number of cells. That's followed by a zone of hypertrophy. Those same cells now start to expand and get really large. Hypertrophy means increase in size. And what's happening is these cartilage cells are actually starting to die off. And they're developing a lot of cytosol within the cells, and they're getting very large. I call them the chubba cells, because they go from being kind of looking like your pancakes all stacked on one another to then these large cells with lots of cytoplasm and a very translucent appearance and spreading themselves out. But they're dying off very slowly and become replaced by bone cells. So then our next region is calcified matrix. This is where cartilage cells have died and are replaced by spongy bone. So now we have bone cells in there. And that produces this beautiful spongy bone appearance. This occurs primarily during adolescence until skeletal maturity is reached. The epiphyseal plate closure, the fusion of diaphysis and epiphyses, occurs generally in females around the age of 18 and males by the age of 21, and then become an epiphyseal line, a closed growth plate. An epiphyseal plate is an open growth plate, which means it's capable of lengthening the bone. So here we're looking at a slide. It doesn't, unfortunately, show you the resting cartilage above, but it would have a very classic hyaline cartilage appearance like we looked at in some of the previous slides. In lab, if you're doing this face to face, you can very clearly see this. So it's showing you in the upper left-hand corner, the specimen, where you're taking this from, a bridge between the epiphysis, the epiphyseal plate, and then the diaphysis. The resting cartilage would be to the above, to the superior aspect. Then it's followed by cartilage going through this process of change. Right, so you have the zone of resting cartilage. The next zone is the zone of proliferation. Right, even though they call it number one, we refer to it as number two. Cartilage cells undergo to mitosis, so increasing in number. Do you see how now these cells, they kind of are all stacking on themselves? You see these vertical orientations of cells over and over again. They're followed by, below that, our zone of hypertrophy. Oh, goodness gracious. Hold on, let me go back to where I was. Okay. The zone of hypertrophy, increase in size. So now look at those cells. You can see a dark purple nucleus. You can see all the cytoplasm around it. They now look like these huge cells, which they are. They're hypertrophic, or they're hypertrophic. So you have a stacked coin cell appearance above it. And now we have what's called the zone of hypertrophy. So that's why I call them the chubba cells, because all of a sudden they're huge. They die off and become replaced with spongy bone. And that's our zone of calcification, ossification below that. The pinkish regions, the light pinkish regions, those are the trabeculae. And the grainy portions there, that's the red bone marrow. They're the two components that make spongy bone. So there are pictures in your other pictures in your textbook. There are definitely pictures in the lab book, and in lab you can actually look at slides of this. For me, that's the area I use for spongy bone, because it's classic looking. All right. So you have those four zones. All right. You have a zone of resting cartilage, zone of proliferation, zone of hypertrophy, and zone of calcification, maybe / ossification. In that order all the time. It might be from top to bottom or bottom to top, but it'll always be in that order. They never change. Okay, but then we need to have appositional growth, and that's the increasing of the diameter of the whole bone. And so we have the activity of the osteoblasts and osteoclasts here. So if we want to increase the width, the thickness of the entire compact bone region around the entire bone, especially along the diaphysis, on the outer surface of the periosteum, we stimulate the osteoblast to lay down more bone and thus start to thicken it, and widen that bone. But when we do that, we want to keep the rim and thickness of the compact bone region rather stable. We don't want it to maybe remain paper thin, but we don't want it to get, as thick as, let's say, the medullary cavity. So we let those osteoblasts do their thing, lay down bone on the outer surface and let it kind of grow from the outer margins. But then at the endosteum, we'll stimulate the osteoclasts, right, to liberate a lot of those lysosomes that they have and dissolve some of that bone there so that the compact bone rim gets thicker, but not really thick, and then it's kind of stabilized, as you reach bone maturity. And thus, when it does that, you build it up on the outer periosteum and you break it down some on the endosteum. It lets the medullary cavity become wider and longer, where you can, again, in childhood, store more red bone marrow there for blood cell production, and be replaced in adulthood with yellow bone marrow. So that is what we call remodeling, when we're building up some areas and breaking down the other. We do that with fractured bone. Osteoclasts go in there and, help break down some of the components of fragmented bone. So do some of the white blood cells, right? And so you break that down, you replace it with some fibrous tissue, and then we send in the osteoblast to lay down some new bone material. And the osteoclast can kind of come in and refine it, kind of like sandpaper or nail file, nail file down all the rough edges, so it becomes looking most like normal bone tissue. Okay, hormonal regulation of growth. Well, we have a couple different hormones that are associated with this. We have human growth hormone, which we look at in the endocrine system in AMP2. It's a hormone that's released from the anterior pituitary gland, and it stimulates the growth of cells at the epiphyseal plate. There are a couple different hormones, of course. So we have growth hormone. Growth hormone stimulates those cells. Growth hormone is also associated with letting cells repair after working hard all day, stimulating protein synthesis and things like that. Thyroid hormone, right? We have a surge of thyroid hormone. Thyroid hormone also helps regulate growth, or proportional growth. It also regulates your basal metabolic rate, like how efficiently your body cells convert substrates like glucose and fats and proteins into energy of the cell to produce ATP in the mitochondria. And then we have sex hormones, and that's a big component of growth spurts, because as you know, when you're entering into adolescence, right, women start to produce more estrogen and progesterone, and men produce more testosterone. And both of these sex hormones for males and females stimulate bone elongation and widening. That's when you start to growth spurt. So you not only, those hormones are associated with developing secondary sexual characteristics, but it stimulates muscle growth, bone growth, fat deposition, things like that. So they all work together. They all have an integral part. Okay, bone homeostasis, the basic process of remodeling bone. We have the osteoblasts and the osteoclasts that work together, but in opposite directions, right? Because osteoblasts build up bone and osteoclasts break down bone. So bone deposition means to lay down more bone. That's done by the osteoblasts. Build up and lay down bone occurs when bone is injured or added strength, or we're trying to, in adults, how to build up a bone and increase the length or the size of a bone in an adolescent. Bone resorption, it's called, is due to the osteoclasts. They resorb or break down bone. They secrete lysosomal enzymes. Remember, the lysosomes were those little vesicles that contain digestive enzymes made by the rough endoplasmic reticulum and the Golgi body. And they have a lot of acids and nutrients, enzymes to break down that compact bone, and allow calcium and phosphate to enter into the bloodstream. So remodeling control, a lot of the remodeling of it is due to hormones that are released from different glands of the body. And this is, your really first true negative feedback loop that you're going to learn. We use this again in the endocrine system in AMP2. And then actually we come back to it again in the urinary system, because we see how parathyroid hormone targets a certain region to reabsorb this calcium. Okay, so we have normal blood calcium levels in blood. Everything that can be found in blood, there's a normal level for it to be in your plasma or in the blood of your body. When it goes too high or too low, some of these, different electrolytes, or let's say in the case of glucose, which is not an electrolyte, needs to be homeostatically, very actively and dynamically balanced. Not everything we will, if it drops too low, we're going to necessarily have a hormone to correct its level and bring it back into homeostasis or in normal levels. But some of these do have them. And calcium is one of them. So this is the first one. All right, when calcium levels are lower than normal, the gland called the parathyroid gland secretes only one hormone, thankfully, parathyroid hormone. And that hormone has multiple actions, because when you're low in things, it's usually more difficult and more complicated in the way you're going to raise it back up somehow and get it back up into a normal range. Because if you had a sufficient amount in your diet, you would be using the calcium that you're eating every day to maintain these levels, but you don't. So when calcium levels fall low or below normal ranges, parathyroid hormone stimulates the osteoclast, right, to resorb bone, to break it down and release calcium and let it enter into the bloodstream. So we start elevating blood calcium levels right there. Then parathyroid hormone, or PTH, targets the kidneys to reabsorb calcium, because the kidneys filter your blood, and excess calcium will flow over and then ultimately be passed out of the body in the form of urine. Well, while you're forming that urine, if there's calcium in there that you could use, then let's reabsorb it and put it back into blood. So that's what parathyroid, PTH will do. It also targets the kidneys to convert vitamin D, the vitamin D that we made in skin, or maybe we have taken in nutritionally, and convert it to calcitriol. Calcitriol is the active form of vitamin D. Vitamin D can travel around your body, has other purposes as well, other uses in your body, not just to be converted to calcitriol. But when you need it, right, PTH will convert it into calcitriol. Calcitriol targets the small intestine to absorb calcium from the food that's passing through your small intestine. Because if you don't, then it's just going to go to the large intestine and go out in feces. So if you could use it, then reabsorb it. And that's what calcitriol does. So when blood calcium levels are low, the method that we correct it is a little more complicated. When it's higher than normal, the thyroid gland, which is found in the throat area, you have the thyroid gland that's below your voice box. It's kind of a butterfly shape. It's found there. If you pulled it off and looked to the posterior aspect of it, you would see these little glands called the parathyroid glands. They kind of look like little marbles stuck to the back of your thyroid gland. But in any event, it's found in the throat region. It releases calcitonin. And calcitonin inhibits osteoclasts, right? So they don't keep breaking down more bone and putting more calcium in blood. There's enough there. And in the same respect, they will stimulate the osteoblast, because if you have this extra calcium, then let's try to take it and store it and build up the bone to be really strong. So when, let's say, blood calcium levels drop too low later, we have a little savings bank of calcium we can pull from without totally demineralizing and creating very fragile and weak bones. So we inhibit the osteoclast, we stimulate the osteoblasts to store as much calcium as possible, build strong bones, and in the process, we decrease the blood calcium levels back to normal. So when calcium levels are high, parathyroid hormone is used. I'm sorry, when calcium levels are low, my bad, we use parathyroid hormone to increase blood calcium levels back to a normal range. And when blood calcium levels are higher than normal, we use calcitonin. Do not confuse calcitriol and calcitonin. Ding, ding, ding. Calcitonin will decrease blood calcium levels. All right, so this just kind of shows you the back and forth, the negative feedback loop, of parathyroid hormone. Fracture and repair, this is really of an FYI, but you can look through this. I don't ask you any questions, but sometimes, depending on who you have for lab and which campus is teaching lab, some of them will get into this a little bit. We personally don't, at the Gibbs campus, don't have an exercise for that unless that particular instructor happens to be using the lab book, which usually does not occur. But when we have a fractured bone, we have what's called the hematoma formation. That is, a hematoma is a bruise. It's where the bone is damaged and the blood is leaving the blood vessels in the center of these osteons, or maybe even tore them as they enter the compact bone. But you have a bruise, essentially accumulation of blood. It's followed with swelling and inflammation. And along with that swelling and inflammation, it brings a blood supply and starts the repairing process of granulation tissue that forms from fibrous tissue. It ends with what's called a bony callus. It's kind of like a scab that's connecting two regions of bone. And then we have bone remodeling. The osteoblasts go in there and start laying down nice, good bone, and the osteoclasts come and degrade some of the bone and kind of smooths out the surface, kind of like taking a nail file to your nail, and then you get it back to hopefully as normal as possible. Fractured bone and healed bone will never be quite the same as the bone you were born with before it was ever injured and repaired. So here we show you a hematoma formation, essentially blood escaping the cardiovascular system, that's bruise. We form a callus, then we have bone remodeling, and hopefully it gets back to as normal size as possible. Every fracture of a bone can be seen on a radiograph. It become a much denser kind of region of bone. Homeostatic imbalances that we want you to know are osteoporosis and osteomalacia. Osteoporosis is bone resorption, is when, I should say, bone resorption exceeds bone deposition, so that bones become more porous and fragile and essentially what we call demineralized. With age, as you get older, it's going to happen with a decrease in estrogen and testosterone. You lose what's called the inhibition or to the osteoclast that you normally had. So let me just kind of explain that. Some of the functions of some of the hormones in your body, like some of the sex hormones like this, estrogen and testosterone and things, they kind of keep a muzzle on the osteoclast so that, even though, we don't necessarily have to, have, like, let's say parathyroid hormone to stimulate them, when we don't have parathyroid hormone, we intermittently use these hormones to keep a muzzle on them until you really need them to break down compact bone and, as a supply of calcium into the bloodstream. So there's normally a certain amount of inhibition to those osteoclasts by some of these sex hormones. But as we get older, those hormones diminish. And now you lose some of that inhibition into the osteoclasts, and they start kind of going a little wild and breaking down bone unnecessarily. We also have changes to the other hormones of the body. I'll just tell you that as you get older, all the glands of your body start to degenerate a little bit and produce fewer amounts or less amount of the hormones that that particular gland is supposed to secrete. They don't necessarily all do that all at the same time. They do so kind of at different pace. And that's what's associated with aging. But in any event, for women especially, during and after menopausal, and as men get older too, because their testosterone levels drop, their bone becomes more porous and subject to fracture. So that's osteoporosis. And that can be due to, it's not necessarily later in life, it can be due to the action of different pharmacology that you're taking, different drugs. Sometimes, when you have had cancer to some of the structures, we're not covering the reproductive system now, but, if you have, ovarian cancer and your ovaries are removed, for men, if they're, they've had testicular cancer, then you are going to have changes to the sources of testosterone and estrogen and progesterone in females. And so, it can change the density of bone, and it does. Osteomalacia is similar, but it's due to a dietary insufficiency of calcium and or vitamin D, and that weakens the bones. When that occurs during adolescence, or as a child, it produces weakened bending bones, and that's called rickets. If this can be identified and rectified early, then as you go, children go through their growth spurt, then you will start building like healthier bone. But if it exists for a long period of time, it can do pretty much irreparable damage to bone. It's very hard to change that after years of existence. But you can have periods of insufficient calcium and vitamin D, and it won't necessarily, have a dramatic effect on the skeleton. But this is why we have things like vitamin D, fortified milk. Why we use, there's a lot of calcium in cow's milk, so we use that as a source during adolescence, and throughout life, really, for calcium and vitamin D. Here on the top, we see the trabeculae of normal bone, and then below that, osteoporotic. So the trabeculae look like walls of the sponge, and they have, the trabeculae has a kind of soap bubbly kind of appearance already, where you would have red bone marrow in between the walls of the trabeculae. But if you actually start breaking down and make each wall of the trabeculae more porous, then it's like Swiss cheese within Swiss cheese kind of an appearance. And that trabeculae, in the normal composition above, actually imparts a very good amount of tensile strength, resist compression, because it travels in multiple directions. So the forces placed on bone from multiple directions can be kind of translated and diffused, so to speak, along the walls of these trabeculae. But if that happens, with osteoporotic bone, they just start to fracture. That takes us to another Kahoot, which I'm not going to do again, because I don't want anything to happen, but we will go through, and that's the end of the PowerPoint. Let me just stop that share. So we will, on Wednesday, I'll go to my office and Zoom the class from there. So hopefully we won't have any problems again. I don't think it's something on my thumb drive here. I just think it's something on my computer. So I apologize for that. And it has less distractions. You don't hear the dogs barking and things. So, okay, so when we meet on Wednesday, we will go back through the Kahoots. And if I forget, please remind me, but I will probably remember. Then we will go through the axial and appendicular skeletons. We're not going, I asked you some of that information on a lecture exam, but it's mostly lab material. So we will probably, I will kind of give you a heads up on the types of things I would ask you. Most of it will be why things named are the way they are, like what is a foramen? What is the difference between a foramen maybe and a canal? It's the bumps and the holes through bone. Maybe what bones will be part of the appendicular versus the axial skeletons. But as we go through that, I'll kind of give you a heads up. And of course, the quizzes help too, because you get 2 tries for that. And hopefully you're getting a variety of questions. There's generally at least 20 to 40 questions in a question pool. So even if you get 100 on one, I would suggest taking it again, because the highest grade sticks. And worst case scenario, you see questions, maybe that you did not see before and you answer it wrong. Well, so be it. But at least you saw the questions and you get to review your quizzes. So it's a good source to study from for the exam. Occasionally, some of those questions, show up, but most of them are going to be newer questions, as you probably experienced in exam one. So as soon as the people who have not taken exam one have taken them, and if they're not completed by probably Wednesday, by the time we're about ready to Zoom again, their opportunity will have passed. I'll just open it up for review for you, because you're probably dying to know what you got right and what you got wrong. Okay, so does anybody have any questions?
No.
Okie dokie. I think now that we're starting this phase, and after the first exam, you get a good feel of how to approach this. So all right then. Well, then I will end the Zoom here. We will meet back 5:30 on Wednesday.
Thank you.
You're welcome. Bye. Sorry about the break in the Zoom there.
Thank you. Have a good day.